We're interested in the question of how people hear the beat in a musical rhythm. We focus here on the main beat, which we call the pulse. Pulses have a particular frequency and phase with respect to a rhythm, and these are assumed to correspond to the movements that people make when they clap or tap along with a rhythm. In the simplest kind of rhythm, called an isochronous rhythm, sounds are equally spaced in time. For a rhythm like this, people almost always perceive the pulse to coincide with the sounds. In a synchronization experiment, people tapped at 2 Hz, and taps were generally in phase. In syncopated rhythms like this one, sounds are not equally spaced in time, but people's claps have the same frequency and phase as with the isochronous rhythm on the previous slide. In the synchronization experiment, people mostly tapped at 2 Hz, and taps were generally in phase. However, some were antiphase, and sometimes subject failed to synchronize. What happens when people aren't moving? Our hypothesis is that, in the absence of overt movement, the perception of pulse is an oscillation similar to the physical oscillation that generates claps. For the isochronous rhythm, the frequency of the oscillation will be the same as the frequency of the sound onsets. From a perceptual point of view, pulse corresponds directly to the stimulus itself. But for the syncopated rhythm, the situation is more interesting. This particular type of syncopated rhythm is called a missing pulse rhythm, because frequency analysis shows many different frequencies, but no energy at the perceived pulse frequency. To test this hypothesis, we asked whether we can detect pulse frequency oscillations in steady-state evoked potentials for missing pulse rhythms. If so, this means the brain must somehow be generating the pulse frequency because it is not present in the stimulus. A pulse frequency response could not represent a series of evoked potentials because a series of evoked potentials would similarly show no energy at the pulse frequency. We can use the missing pulse property to distinguish areas that respond mainly to the physical stimulus from those that generate the pulse frequency. We have published a dynamical systems model of this process. A series of sound onsets flows from brainstem into an auditory cortical network where it synchronizes oscillatory populations at multiple frequencies. These oscillations then stimulate a motor planning network where different oscillatory populations give rise to the pulse frequency. Why a motor planning network? Empirically, it has been known for some time that listening to musical rhythms recruits motor regions of the brain, such as premotor cortex and supplementary motor area. However, imaging technologies like PET and fMRI do not have sufficient temporal resolution to tell us what's happening in these areas. Patel and Everson's action simulation for auditory prediction, or ASAP hypothesis, explicitly predicts that information flows along the dorsal auditory pathway and that pulse perception takes place in motor planning areas of the brain, where it is realized as simulation of periodic action. Our neural resonance model shows how this process could be embodied in a multi-frequency dynamical system consisting of 2 oscillatory neural networks stimulated with a missing pulse rhythm. In 2 EEG experiments, we asked whether we could measure SS-EPs in response to missing pulse rhythms and localize the neural sources of the pulse frequency. Both experiments used the stimuli from an earlier study, an isochronous control rhythm, a random control rhythm, and 2 missing pulse rhythms. In this study, however, we used a different task. We asked subjects to listen to the rhythm and prepare to tap the beat, which we called the perception phase. Then, after the warning tone, they were to tap the beat, the synchronization phase. This task has 2 important features. It forces subjects to imagine the beat during the perception phase, and it enables us to know whether they heard the beat or not, and if so, whether it was in phase or antiphase. We only measured SS-EPs during the perception phase, only for those trials in which they successfully heard the beat. Our model can predict SS-EPs. The model auditory network produces strong energy at 2 Hz for the isochronous rhythm, but only minimal energy at 2 Hz for the random control and the missing pulse rhythms. On the other hand, for the missing pulse rhythms, the pulse frequency is evident in the motor planning network. In experiment 1, we first considered the synchronization phase. In the missing pulse rhythms, people mainly perceived a pulse at 2 Hz, occasionally at 1 Hz, and mostly in phase. But sometimes in antiphase, and in some trials, people did not perceive a pulse. Using this information, we computed SS-EPs for the perception phase. In the grand average over all electrodes and subjects, we see significant energy at the pulse frequency for the missing pulse rhythms and isochronous control. We saw a typical auditory topography for the isochronous control, whereas for the missing pulse rhythms, the 2 Hz topography suggests different neural sources. Experiment 2 used a 256 electrode EGI system and structural MRI scans to identify regions of the cortex in which 2 Hz energy accounted for a significant portion of the source space time series. We projected the sensor time series onto 60,000 orientation-constrained vertices on the cortical surface. Working in source space, we computed SS-EPs for each vertex and normalized before taking the mean over subjects. Finally, we used the Brainstorm software to visualize areas where the 2 Hz amplitude exceeded a threshold in at least 30 contiguous vertices. Note that sources are different in the missing pulse and isochronous conditions, and no significant sources of 2 Hz energy are seen for the random control. We see areas consistent with both dorsal and ventral auditory streams. In the missing pulse condition, we identified sources in bilateral Brodmann area 6, including both SMA and PMC. We also found bilateral inferior frontal gyrus and anterior cingulate cortex. We also identified superior temporal gyrus on the left and supramarginal gyrus and M1 on the right. Note also that no pulse frequency source was observed in primary auditory cortex for the missing pulse condition. This was expected because the stimuli contains no energy at this frequency. Here we show the full SS-EPs for regions of interest that were defined based on the previous analysis. Note that they all show strong energy at 2 Hz, and most of them also show strong energy at 1 Hz, a frequency that's also missing from the spectral analysis of these rhythms, but which participants identify as the pulse on some trials. Finally, we see that in the missing pulse condition, 2 Hz energy was significantly greater than 0 in most of these areas. However, in these regions, 2 Hz was almost always stronger for the isochronous rhythm, and it was never significantly different from 0 in these areas for the random rhythm. In summary, we used an explicit motor imagery task in skilled subjects and found strong responses at the missing pulse frequency. The missing pulse property enabled us to identify cortical sources of the pulse frequency separate from stimulus frequencies. Pulse frequency activity was observed in both dorsal and ventral auditory streams. In the missing pulse condition, we found extensive activation of SMA and PMC, as well as STG, SMG, M1, and ACC. Our next steps will include a new experiment using the beat alignment task, which does not require explicit motor imagery. We also plan to refine the model based on the results of these experiments.